Hello friends, and welcome again to mechanistic pharmacology. Friends, today we are going to start with the new lecture series content possesses a title antiviral agents, which is based upon the PCI syllabus. Now, in this lecture series, there are total number of 4 lectures. First lecture is introduction of virus, which we are going to discuss now, followed by lecture 2 and lecture 3, that is antiviral agents, and lastly very important lecture number 4, comprised of exercise and multiple choice questions with the reasoning answers. So friends, today we are going to learn with the lecture number 1, that is introduction of virus. Thank you. Viruses. Friends, viruses are called as intracellular parasites, that is they contains any one kind of nucleic acid. Either they contain DNA or RNA, but never contain both. Friends, viruses are also called obligate parasite, in other words, because for their replication process they need metabolic process of host cell, as because they do not have necessary enzyme for protein synthesis or either nucleic acid synthesis. Friends, viruses do not have cellular organism, that is why they do not fall under class of unicellular organism. And unicellular organism, they contains both kind of nucleic acid, either DNA and RNA, and synthesize their own macromolecule. But viruses contains either DNA or RNA, but they never contain both. They direct host cell to make new viral proteins, and in such cases they are unaffected by antibacterial antibiotics. Based upon their classification, they are DNA containing virus or RNA containing viruses. Now, example of RNA containing viruses is rotavirus, which cause diarrhea, enterovirus responsible for polio, paramyxovirus for measles and mumps, orthomyxovirus causes influenza, retrovirus causing HIV/AIDS. Friends, these are the examples of RNA containing virus. Now, examples of DNA containing virus is adenovirus causing respiratory or eye infection, pox virus causing smallpox, cytomegalovirus causes chickenpox, herpes virus causes herpes simplex, and hepatitis B virus causes cancer. Common structure of virus and their function. Friends, viruses contains envelope proteins or glycoproteins, or in other words we can say surface proteins. Now, the function of this is that they are responsible for attachment, means through envelope protein or glycoproteins or surface proteins they attach with the host cell. They also contains membrane protein, which is responsible for the process of fusion. It means when viruses attach towards the host cell, the membrane protein of virus is fuses with the cellular membrane of the host cell, and they releases the kind of nucleic acids into the host cell. So, this is the function of membrane protein, which is responsible for the process of fusion. Lastly, they contains genome, as I stated, either DNA or either RNA, as well as nucleocapsid core. Now, this is responsible for the process of replication, means as soon as the viruses attach with the host cell, the cell membrane protein fuses with the host cell membrane, and finally the genome, either DNA and RNA, is introduced and the process of replication, that is transcription or translation, is followed. And that is why these viruses, they replicate inside the host cell and they use the metabolic energy during the process of host cell for their replication. Now, replicative cycle of virus. Friends, now consider this is the hypothetical structure of host cell, now where the virus replicate and it synthesize new viral particles. In this process, I am going to depict the replicative cycle of virus through which they produces or they synthesizes new viral particle by using the host cell energy. Now let us consider this is a hypothetical structure of virus which contains either DNA or RNA. So, the life cycle of any virus depend on the first process called as adsorption, means here the virus adsorb into the host cell via certain glycoproteins present on their cell surface. Now, this process is called as attachment or entry, and via process of endocytosis they enter inside the host cell, means the process of penetration begins here and the membrane protein of virus fuses with the cellular membrane of the host cell and they enter inside the virus. Now, after it the process of uncoating begins, means virus causes introduction or transfer of their genome, either DNA or RNA, so that they can produce or synthesize new viral particles. Friends, during this process they utilize the energy of host cell, they utilize the synthesizing enzyme of host cell, so that they can produce new viral particles. Friends, they do not replicate inside the host cell, but also they direct them to make new viral particles, means here this early transcription or via messenger RNA they synthesize new enzymes, new viral particles via ribosomal structure. We all know that ribosomes, they are responsible to synthesize new proteins. So, when the genome is uncoated inside the host cell, now these viruses, they are smart enough so that they can make new viral proteins via ribosomes of host cell. Now, during this process early transcription, early translation begins via certain enzyme and synthesis of new viral proteins begins, and new viruses or new viral proteins or virions, they again assemble and the structural viral proteins forms. Now, after this exocytosis process causes the virus to come outside the host cell, and after this they replicate in a numerous number, numerous structure, and the release of virions causes to infect other host cell. Now, challenges against viral infection. Now, the first challenge is the development of drug resistance. Friends, as we continue the treatment via antiviral agents, the drugs are prone to be resisted by the host machinery, or we can say the viral assembly. So, the development of drug resistance is very important challenge against the viral infection. The next challenge is narrow antiviral spectrum. Friends, few drugs have a very narrow or limited antiviral spectrum, means these drugs, of course they are antiviral agents, but due to the proliferation rate of virus, these antiviral agents, they possesses narrow antiviral spectrum. So, to enhance the spectrum, we can either use the combination therapy such as we are using in HIV AIDS. So, against the human immunodeficiency virus, as we all know, the proliferation rate is much higher, and therefore, the combination therapy of 2 or 3 drugs or 4 drugs is used against HIV infection. The next challenge is toxicity due to varied dose. Friends, as we all know that the multiplication rate or proliferation rate is very high. So, researcher want the drug to be in such a dose so that it can inhibit the proliferation rate as well as it can kill the virus as early as possible, and this is the reason why the toxicity introduced, because as the dose varied, toxicity causes, and therefore the antiviral agent possesses number of toxicities. The next challenge is proliferation rate. As I said to you that the early transcription and early translation process of the messenger RNA begins and synthesis of viral DNA or RNA forms. So, here during this process the budding or release of virus is at much higher rate as compared to the normal rate of the host immune system. So, during this process antiviral agent should be in a sufficient concentration so that they can combat with the high proliferation rate, or budding or release of viruses should be inhibited as early as possible. So, the proliferation rate is again a big challenge for the infection. Now, altered immunological response or reduced susceptibility of the host. As we all know, the immune system of everyone is differs. Therefore, the immunological response to fight against the infection of every human being is different. So, this is also one of the challenge for the viral infection. Now, importantly, lack of understanding about the path of replication. As we all know, friends, that during the process of replication, the viruses followed 8 steps, for example, adsorption, then penetration, then uncoating, then viral assembly, budding or release of virus. So, during this replication process, many viruses and they uses host enzymes, host proteins to, for their process of replication. So, this is the biggest challenge to find the path of replication, how these viruses they replicate, what the enzymes they are used, what type of structural proteins they are forms. So, this here the lack of understanding, or we can say the pathology, pathophysiology and path of replication should be understood so that we can use the antiviral agents. So, lastly, the lack of understanding is very essential to know the path of replication, and this lack of understanding continues to begin as we are seeing in the COVID era. Now, friends, lastly, time and strength of therapy. As I stated that the drug should be at sufficient time, should be at sufficient duration so that it can combat with the high proliferation rate of virus, and the strength of the therapy means the antiviral agent should be used in combination so that the replicative cycle of virus should be inhibited by the strength of therapy. Now, goal of treatment. Now, up till now we had seen that viruses, they are the smallest unit, they are the smallest infective agent that uses the host cell machinery, and they redirect host cell to make new viral proteins. So, friends, the goal of treatment should be like that few agents that either inactivates the intact virus, which comes under category of virucides. So examples of virucides is detergents, organic solvents or ultraviolet rays. Now another goal of treatment is that these agents should inhibit the replication process of virus at cellular level. So for this process, for this inhibition, antiviral agents are used to inhibit the replication process of virus, so that these antiviral agents, they do not they inhibit the process of replication and they inhibit the synthesis of new virions outside the host cell. Examples of few antiviral agents like acyclovir, foscarnet, ritonavir. Now another goal of treatment that modulate the host response towards infections, and the category is immunomodulators, means we can modulate the host immune system so that the host cell or human being, they are unaffected by the viruses. So for this purpose, modulators such as herbal remedies, serum antibiotics or interleukin 6 inhibitors are used. So this is the goal of treatment, means when the virus introduced inside the host cell, what should be the goal of treatment. So first goal of treatment, either we can inhibit or either we can inactivate the intact virus by using virucides, or either we can inhibit the replication of virus at cellular levels. So for this, the category of antivirals is used, or we can finally modulate the host immune system towards the infection. So for such cases, immunomodulators are used. For examples like herbal remedies, serum antibiotics and many more. Now coming towards summary. Friends, as we stated that these are very tiny infectious agents, they enclosed in a coat of protein which is called as capsid, and inside it they contain either RNA or DNA, and they require our body's energy process to invade or multiply or replicate. Friends, they spread via droplets, through coughing, sneezing, stools, physical contacts or via aerosol routes. So some are contagious but many are not. So friends, for their prevention process, what should be the goal? So first is to maintain the good hygiene habits. Wash the hands frequently and maintain the hygiene of the person. Secondly, use the antiviral agent so that the process of replication can somehow be limited. And finally, the vaccination is the last source through which we can keep away from these virus. For example, there are multiple type of vaccines are available like BCG vaccine, influenza vaccine and many more type of vaccines which can combat throughout the life against the viral infection. So friends, here we end with the introduction of virus. In the next session, we are going to learn about the antiviral drugs. So stay tuned. Thank you very much.